We'll be at our next Science Night Live event, which will be coming up on March 8th at 7 p.m. Dr. Stoffen is former science director at NASA, and she's coming here to help us kick off our celebration of 50 years of Apollo, which will culminate with a conference in the summer of 2019 on the year since the Apollo landing. So we're very excited to have you here, such a well-known scientist here to kick us off. Dr. Stoffen's speech talk will be in this room, as I said, March 8th, 7. But today we're to celebrate the life of Charles Darwin. Now, Charles Darwin was born on February 12th, 1809, and people have been celebrating his birthday since he was born. So his parents' party, but we continue to celebrate his birthday today. However, the regular annual celebration of Darwin Day didn't really take off until the 1990s. So all across the world during February, there is a celebration at many universities, schools, scientific organizations about Darwin. Now, why do we celebrate Darwin now, so long after his birth? In part, it's because science has been under attack. Whether we look at evolution or climate change, some other topics, we have to remain vigilant to those, to look out to those who deny or suppress the truth. Now, of course, Darwin himself was widely attacked during his life, but he persevered, and his ideas live on. He serves as an example to all of us interested in science. Before I introduce the speaker, though, I would like to ask if I could have Charles Darwin himself come out and say a
Ladies and gentlemen, welcome. Most kind of all of you to make such a fuss, man. Most kind. May I also say that it is a great pleasure to be invited to this auspicious university to provide a few opening remarks. After all, how often is it that one is asked to deliver one's own eulogy at one's own birthday? I was born Charles Robert Darwin on February 12th, 1809. I am told by my host each year on or around my birthday, events like this are held to honor my life and work. I'm very proud of my accomplishments as a geologist and of the explorations of South America I made traveling the HMS Eagle. My greatest satisfaction comes from being recognized as scientists carefully documented the evidence for evidence, discovered a mechanism to explain everything. My idea of natural selection. My theory of evolution was published in 1859 in On the Origin of Species. It is no doubt the chief work of my life. And it was from the first highly successful. The initial small edition, 1,050 copies, was sold out the day of publication. The second edition of 3,000 copies sold out after. 16,000 copies sold in English. And considering how stiff a book it is, that's a pretty good sale. The final chapter on the order, I wrote, in the future, I see open fields far more important. Much light thrown on the origin of man. Later, in my book, The Descent of Man, I made explicit scientific case for humans being a product of biological. Of course, this upset many people who objected to the idea that humans were not specially created by God. I never wanted to upset anyone. the evidence for the evolutionary origin is overwhelming. Soon see. Tonight, we are very fortunate to have Dr. Anyway, tonight, we are very fortunate to have Dr. Robert Hill provide us his insight into the evidence of human evolution through the lens of those most extraordinary of creatures, dinosaurs. Dr. Hill is a vertebrate paleontologist who specializes in the study of armored dinosaurs, the Thyreophora. He is associate professor of science education, director of the Anatomical Gift Program of the Donald and Barbara Zucker School of Medicine at Hofstra. Dr. Hill attended Hofstra as an undergraduate study biology before moving on to graduate study at Stony Brook University, where he earned a doctorate. Please join me in thanking Dr. Hill for generously volunteering his time to speak on the topic form, function, fossil, and physician, surprising evolutionary connection between dinosaurs and human anatomy.
Thank you. Thank you. Wow, Charles Darwin, everybody. Doesn't he look great for 209 years old? Well, thank you very much for that great introduction, and thanks to everyone for being here tonight. And especially thank you to the Hofstra Cultural Center for inviting me to give this talk. It's a real pleasure to address all of you in this room in particular, because it was right here in this room, see that seat right there, that I attended my first lectures in biology as an undergraduate here at Hofstra University. And so to be able to work here and to be able to lecture from this stage is really like coming home. Now, I've been fascinated with the structure of the structure of living things from a very early age. And one of my earliest memories is being in my parents' basement, looking through this book, and I just love these whimsical illustrations by the illustrator Cornelius DeWitt. You can see that he really matches anatomical accuracy, like the bones in the brain there, and pairs it with some of the functions in a whimsical way. Like you can see the intestines as pipes and the heart, the mechanical pump. And I remember in particular looking at one page, page 23, you can come up and look later, and there was a picture of a human skull, just like this one that I have here. And next to it was this jumble of bones that I couldn't really recognize. And I asked my mom, what's going on here? You know, what happened to the skull? Why is it broken up like that? She said, well, the skull is not just one thing. It's made up of multiple bones all connected together. And I was fascinated by this. I wanted to know more. And she said, well, not only that, but each one of these little bones has a name. And I was floored. I said, I have to know all the names of these bones. And then she said, well, not only that, but each little hole, each little bump on those bones has its own name. And I was completely blown away. I had to figure out all the relationship to these things, and I'm still working on it now. Some of my students can attest. What I didn't know then, but I understand now, is that I was making my first forays into exploring what biologists call the hierarchy of structure. And what this means in a nutshell is if you take two or more of something at a low level of organization, you can put those things together and make something that's a little bit more organized. So, for example, if you take two or more types of cells, that gives you a tissue. Two or more types of tissues gives you an organ. And if you put two or more types of organs together, working towards a common function, well, then that gives you an organ system. All the organ systems put together give you an organism. And so this was something that I was really fascinated with from an early age. But I soon found out that the hierarchy of structure doesn't stop there. It keeps on going. And so organisms come together in populations, species, and higher taxa. And some of those taxa don't even exist on Earth anymore. So that's where this book came in. And I realized I had a lot more to do to understand the hierarchy of structure. So what I hope to do over the next hour or so is to weave a common thread between these two things, my love of human structure and my love of dinosaurs, and hopefully to show you that they really aren't too different from one another. Another person who was fascinated with biological structure from an early age is Charles Darwin. And I love this picture because we often see Darwin depicted in middle age or as an elderly stage of his life. And I love this picture because it shows Darwin as just a happy, unself-conscious child. He's hugging his little plant to him, which he undoubtedly collected or maybe grew it from a seed. Darwin was fascinated with the natural world, and he collected plants obsessively and collected pebbles and insects and was a great naturalist even at the age of 8, where he's depicted here. And he was born to a very well-to-do family. And so this was a hobby, a luxury that he could afford, to have naturalism as a hobby. But the idea was that always he would go into the family business, and that was medicine. So at the age of about 16, much earlier than our medical students start here at the Zucker School of Medicine, Darwin started apprenticing and started his studies to become a physician. And he absolutely hated it. He couldn't stand being in medical school. One of the biggest problems that Darwin faced was that his anatomy lecturer was really, really boring. See, some of my medical students here, I know you guys don't have that problem, right? But Darwin just couldn't stand. He found the lecturer of anatomy so dull and just couldn't pay attention to his anatomy lecture. Another problem that he had was that he couldn't stand the sight of blood. And so when he observed a few surgeries, one of them on a young child, he just about passed out at the sight of not the sight of blood, but at the brutality of surgery performed in an era before anesthesia. So Darwin came back to his home, and he said, "Listen, family, I'm not going to become a physician. It's not in my heart." And his father said, "Well, that's okay. I guess there's another thing that a country gentleman can do. You can become a priest." And Darwin went to school, to the seminary, to study to become a priest. And he didn't take very much to that any better than he took to medical school. He kind of got into partying and drinking and just not spending as much time on his studies as he could. So when he came home, father said, "Well, what am I going to do with you now? You know, you're not going to become a doctor. You're not going to become a priest. What are you going to do with your life?" And it was a series of fortuitous coincidences that led him to the opportunity to sail around the world on the Beagle, which he recounted in his diaries of the trip, Voyage of the Beagle. Now the ship sailed around the Earth for 5 years, and Darwin spent most of that time on land, actually exploring inland South America, and famously landed on the Galapagos Islands, where he observed anatomical differences between different finches that lived on each island. He found on mainland South America exquisite fossils of giant armadillos, and this started affecting his thinking about organisms that had lived on Earth, left their fossils behind, but yet nothing like them found anywhere on Earth today. And again, famously, he landed on the Galapagos Islands, where he discovered that all the individual finches seemed to be adapted to their particular environment and their particular preferred food type. So this was the beginning of Darwin's really revolutionary way of thinking about how species are related to one another, how they evolve, how they change over time. And again, this was famously published in his book, The Origin of Species. Now, what's the number one word that you think of when you hear Charles Darwin's name? Evolution. His name is essentially synonymous with evolution. But Darwin himself did not really use the word evolution very much in The Origin of Species. The term that he did use was natural selection, again, as a mechanism for evolutionary change. And there are really four tenets of the theory of natural selection Darwin put forth. First is the idea that more organisms are produced than could ever possibly survive. If you walk out on an autumn day and just look at all the acorns scattered all around you, if each one of those grew up to be a mighty oak tree, there'd be so many oak trees that you couldn't even navigate between them. So more organisms are produced than could possibly survive. Some of them have to not make it. Second idea is that individuals exhibit variation. So they're not all going to be the same. Some of those acorns, if you will, might be more resistant to cold. Some of them might have a different coloration to them. Some of them might taste a little bit bitter, so a squirrel doesn't want to eat them. And these variations then can be inherited. They can be passed on from generation to generation. And then finally, and this is the crux of Darwin's theory of natural selection, that those organisms with variations that better suit their environment are more likely to survive. Now, another elegant idea that Darwin developed in his book on the origin of species, but he's less famous for, is the idea of descent with modification. Darwin was noticing things about the natural world, such as the similar framework of bones in the hand of multiple different types of organisms. So Darwin noticed, and other scientists of the day were noticing, for example, that in your own upper arm, you've got one bone. Forearm, you've got 2 bones. And then in your wrist, wrist and hand, you've got about 27 bones there. And he noticed that same pattern in dogs and the wing of a bird, flipper of a whale, I think you mentioned more here, wing of a bat, fin of the porpoise, leg of the horse. And this pattern repeated over and over again in the limb or the arm of the vertebrate was intriguing. He also noticed the same number of vertebrae forming the neck of the giraffe, the long neck of the giraffe and the stout neck of the elephant, and even your own neck has the same number of vertebrae, 7 vertebrae in all these organisms. And these facts and innumerable other such facts, Darwin said, at once explain themselves in a theory of descent with slow and slight successive modification. The real revolution of Darwin's thought is here, and this is the only picture that was published on the origin of species. There's only one figure in that whole volume, and what it shows is really the first phylogenetic tree, or the first depiction of what Darwin called the tree of life. The idea here really ran counter to the prevailing view of the day, which was that species were fixed in their characteristics. Now, some people believe species were divinely created. Others believe they just kind of appeared out of nowhere. But what most people agreed in the day was that once those species were there, they didn't change at all. Some didn't even believe that they could go extinct. They thought they just continued on forever. What Darwin was suggesting was that an ancestral population of a species could give rise to multiple different species over the course of generations. Now, this is the figure that Darwin published, but it appeared even earlier in his notebook. And he wrote, I think, drew what really is the first evolutionary tree. He was trying to get at the relationship between closely related organisms, maybe B and C here, and more distantly related organisms over here, indicated by A. And if you know a lot of evolutionary biologists, you probably know one who has a tattoo of this somewhere on their body. Very nice summation of the evolutionary tree there. And you know, over here in Gittleson Hall, I saw it scrawled on a wall. Now, right in the stairwell there. Now, I just thought that was, just somebody spraying it on the wall. But now that I know Charles Darwin's here in the room, I think we might have the graffiti artist right here. But one picture that Darwin would not claim as his own, this one right here. That's also painted on the wall in Gittleson Hall here. And this is one that's often wrongly associated with Darwin's persona. What it shows is kind of this steady march from a chimpanzee up through sort of caveman creatures and then an upright walking man there. And this is one that people often use to level criticism at Darwin's theory by saying, you're trying to tell me that we're evolved from monkeys, then why are there still monkeys living around today? Well, Darwin would counter that this is not his idea at all. And this cartoon by paleontologist Matt Bonin might help to explain that a little bit. Darwin was interested in the common ancestry of all species. His idea is that all species and all organisms on the Earth share a common ancestor at some point back in time. So if you look at this top row here, certainly you are descended from your father. He's descended from his grandfather, and he's descended from your great-grandfather. That is the lineage to which you belong. But that's not your family tree. That's just one way to trace your lineage. Your family tree looks a lot more like this. You can see this lineage here leaves out a big part of your family tree. A better way to depict it is saying you are closely related to your sibling because you're descended from the same parent. You share a common ancestor. You're less closely related to your cousins because you share a more distant common ancestor. Even though it's your grandparents, they're relatively more distant. And so on. You share your great-grandparents, second cousins, and on and on and on through the generation. Darwin's idea was that this held not just for a family tree that you might draw your own pedigree for, but it held for all species that have ever existed on the Earth. So we can use the same model here and say, this is not evolution. No one is saying that fish somehow turned into a salamander, turned into a cat, and that cat turned into you, as the criticism's often leveled. What Darwin was saying is that mammals, like myself and my cat, Sully, we're both derived from a common ancestor, the common ancestor of all mammals. We actually probably share an even more recent common ancestor, the ancestor of mammals. And that Sully and I are closely related to amphibians, not really closely related, but we do share a common ancestor with them. We're certainly more closely related to amphibians than we are to fishes, with whom we share a really distant common ancestor of all vertebrates. So I hope you can see that. I really love this cartoon. I hope you can see how we can extrapolate from just a family tree that you would draw for your own family to an evolutionary tree. But what's the big difference between these? What do you notice? What's the element that really makes them different? Time. How much time has passed here between your parents, grandparents, and great-grandparents? Maybe 150 years, 200 years? A long time, many lifetimes, but it's still a flash in the pan in terms of evolutionary time, age of the Earth. The common ancestor of mammals lived over 100 million years ago. The common ancestor of four-legged vertebrates lived many more hundreds of millions of years ago. And our common ancestor of all vertebrates, nearly 500 million years ago. So this is deep, deep time. It's time on the scale of which we don't think about every day. You're running late for a meeting, and oh my gosh, I'm 5 minutes late for that meeting. Nothing. And years are nothing compared to the span of deep time. So how do we start to think about deep time? Oh, happened to Stegosaurus's head. All right. Well, we tend to lump dinosaurs together as if they all existed and coexisted on the Earth at the same time. You buy play sets and books and things like that have every dinosaur just lumped in one beautiful tableau. But you might be surprised to know that there's less time between T-Rex and an iPhone than between Stegosaurus and T-Rex. Stegosaurus lived 82 million years before the first T-Rex ever appeared. And the last T-Rex left the Earth at the Cretaceous extinction around 65 million years ago. So less time has elapsed between you and T-Rex than elapsed between Stegosaurus. Okay. This is just one example of how vast evolutionary scales of time are. That's really ample time to allow for these transformations between species such that you don't even recognize the ancestral population anymore. The third idea that Darwin put forth in his Origin of Species, which again he's less famous for, but it's nonetheless elegant, is the idea of exaptation or pre-adaptation. Darwin didn't use this term. It was actually coined by the paleontologists Elizabeth Verba and Stephen Jay Gould. But Darwin did first put forward the idea that in considering the transitions of organs, it is so important to bear in mind the probability of conversion from one function to the other. Over this span of deep time, not only could structures change appreciably so that they look different, but their functions might serve a completely different purpose than their original function did. Now, the classic example of this is shown here with these two different bird species. The origin of feathers is sometimes confused or conflated with the origin of flight. But the first feather that ever appeared on Earth certainly wasn't a good flight. We now have lots of evidence of feathered dinosaurs that are not birds, and some of them might be covered with a fine fuzz that might have kept them a little bit warmer. Or they might have had a display function. Even if they weren't brightly colored, they could serve to signal other organisms to come closer or to stay farther away. And so the origin of feathers probably came about for a completely different purpose than what they're used for now. Now, they're really good if they're in the right shape, in the right location, on the right bird. They're really good for flight. Another classic example of an exaptation, or a structure that is pre-adapted for a function that it serves later on, is the branchial arches or the gill arches. What these are, if you have a fish in an aquarium at home, look at your fish. Behind his little eye, there'll be this semicircular slit. And every once in a while, it'll flare out a little bit, and you can get a peek under that. That's the gill cover. And underneath that are multiple arches that wrap around where the neck of the fish would be. Fish doesn't really have a neck, but where the neck of the fish would be. If you like fishing, you might catch a fish like this, and you could lift up gill covers and see the gill arches there underneath the gill. So what are gills good for in a fish? Breathing. That's how the fish breathes. Draws water in its mouth, flows across the gills where gas exchange occurs in little blood vessels, and then comes out through the gill cover. Now, the earliest fishes probably looked something like this thing out of A Nightmare. This is a lamprey, and you can see it doesn't even have any jaws. It's just kind of a wide open mouth and gill slit behind that. And the earliest fishes probably looked something like this. No jaws, just a bunch of gills. Those gill slits are reinforced by skeletal bars made out of bone or cartilage, and there were little arteries running along each one of them. And throughout millions of years of evolution, the first pair of those gill arches became useful at the front end of the organism as a primitive kind of a grasping jaw. And then if we can further add it into a more functional jaw with some teeth, it becomes a really formidable hunting weapon. This is a key evolutionary innovation to have jaws, because once you have jaws, essentially you have hands, because you can grab things out of the water column, and you don't need to just swim around with your mouth open like lamprey. And so the gill arches were functionally useful for respiration in fish. Darwin had a lot to say in The Origin of Species on the branchial arch. Again, branchiae are gills, so anytime you see that word, it refers to gills. Darwin said, in the higher vertebrates, the branchiae have wholly disappeared. But in the embryo, the slits on the sides of the neck and the loop-like course of their arteries still mark their former position. So if you look at these little embryos here, Darwin was interested in what the embryo had to say about evolution as well. If you look at all these little embryos, probably hard to tell. I don't know if you could tell me which one of these is a turtle and which one of these is a chicken and which fish and which one's a human being. They all look pretty much the same. They all look like a little worm with an eyeball and a tail and little gill arches right behind the eye. Well, each of these embryos follows a different path of development. And even though they look very much similar at an early stage of development, they've changed a lot by the time they're fully grown. And so you can see that the fish here, maybe the salamander, we can leave that one out, those would be the only ones that really retain those gills. And as Darwin observed, those slits are there on the side of the neck, but in the higher vertebrates, like reptiles, birds, and mammals here, those slits have completely disappeared. Feel your own neck, you've got a pretty smooth neck. So what becomes of those bones that are supporting the gill arch in these animals which purportedly have a smooth neck? Well, they become a set of bones or cartilages that support the floor of the mouth, they support the tongue, and they wrap around into the muscles of the throat. There are branchial bones or cartilages in all of these different reptiles. And that includes this bird. And they're just about as diverse as the organisms that possess them. So you can see here this snake has branchial bones that look kind of like a pair of tweezers. And this Gila monster here has this bizarre kind of starburst-shaped set of branchial bones. And our crocodile has got this shovel-shaped plate of cartilage with bones sticking off. And then the bird here has this perfect arrowhead shape, kind of stuck on boomerang. I want you to remember that shape. We're going to come back to it later. But this perfect arrowhead is characteristic of the branchial bones of birds. Now, if all of those living reptiles have such a diverse type of branchial bones, surely there must be something in the fossil records that points to what the branchial bones of dinosaurs were like. Well, sadly, it's been disappointing. This is about as good as it gets for the branchial arch of dinosaurs. Each one of these fossils is about the size of a pencil, and they come in two varieties, curved and slightly less. There's not much information that a paleontologist can glean from these structures. They're not rich in anatomical features. So the answer to any problem like that is to find more fossils. Why don't we have good fossils of branchial arches, branchial bones for dinosaurs? There's a couple of possibilities. One is that they're mostly made out of cartilage, and cartilage doesn't fossilize really well. Bone fossilizes okay, but cartilage doesn't fossilize too well. Another possibility is that there's just nothing there to be found. There are no derivatives of the branchial arch of dinosaurs. And a third possibility is that they're there, but nobody's really recognized them for what they are yet, or otherwise found them in the fossil record. So if you want to find fossils, you have to go to where the fossils are. If we were to go outside right now, here on Long Island, it's a pretty bad place to find fossils. There's a lot of ground cover, roads, buildings, and also forest and grass, things like that. If you want to find fossils, you have to go someplace where the rocks are exposed, and the rocks have to be of the right age for the fossils you're looking for. And a great place to find dinosaur fossils is in Central Asia. This is a picture of some outcrop of rock in Western Mongolia. And in 2011, I was really fortunate to be invited to go on an expedition with the American Museum of Natural History to Central Mongolia to look for fossils. Mongolia is an incredibly vast place. You can see how big it is here superimposed on a map of the USA. It stretches from Cincinnati to Salt Lake, or if we want to tip it diagonally, we can go from Long Island down to about Dallas. It's an enormous country, yet very sparsely populated. The same number of people who live here on Long Island in Nassau, Suffolk County, about 3 million, live in the entire country of Mongolia. And half of them live in the capital of Ulaanbaatar. The rest of them live in the outlying countryside. And so you can drive for a long time in Mongolia without seeing another person. Very sparsely populated. It's a land of incredible vistas like this. And in the north, there are these rolling hills. You can see some of the traditional houses of the nomadic people living up there. And in the south, it's dominated by the vast Gobi Desert, where sometimes there's sandstorms. We had a sandstorm blow in on us here. But all of that dust in the air makes for a really spectacular sunset. It's an incredible place to work and to explore. And we spent many, many days exploring in badlands like these, kind of looking in and throughout these little ravines for evidence of fossils. Now, when you find a fossil, it's just about the most exciting thing in the world. You immediately want to dig in and get your hands on it. You have to strain that impulse because one of the worst things you can do is try to prepare the fossil in the field. It just ends up done. What you have to do instead, here I am with a bit of a dinosaur bone that we found right along that outcrop. First, you got to decide if it's something worth taking home. When I first started studying fossils, I thought, that doesn't make a lot of sense to me. Every fossil's got to be worth taking home, right? Well, there's fossils and then there's fossils. So the professionals know what they're looking for, and if you find something that's worth taking home, then you got to carefully excavate around it. So here we are just kind of trying to find out the extent of the fossil. How big, how deep does it go into the hill? Is there a safe place that we can excavate around it and remove it from the rock? And once you dig all the way around the fossil is kind of right here on this top surface, you can start to protect it with a plaster jacket. And what's not shown here is the last step, that little mushroom that now we've made out of plaster, using the same kind of plasters that a doctor would use to set a cast. Now we have to undercut that little mushroom, flip it over, and put more plaster on the bottom of it so it's completely sealed. Plaster cast. And then once you do all that, all your friends abandon you and you have to carry it back to the truck by yourself. But this is a fossil that we brought back, and among many, it hasn't had the chance to be prepared yet. It's still sitting at the American Museum awaiting preparation because there's so many fossils that come out of the rich fossil lands of Mongolia that they haven't all been prepared. Now, in participating in this expedition, we were really following in the footsteps of Roy Chapman Andrews, who, as a former director of the American Museum in the 1920s, he led some of the first expeditions to Mongolia. He was looking not for dinosaur fossils, but for the remains of ancient humans. And one hypothesis at the time was an out-of-Asia hypothesis, that the origin of modern man began in Asia. So Andrews was interested in tracking down that lead. And it's also commonly said, although George Lucas has never substantiated this, that Andrews was the real-life inspiration for Indiana Jones movie. So even if it's not true, George Lucas hasn't weighed in on it, but you can see where that idea comes from. So Andrews didn't find the evidence of early man that he was looking for, but he did find some incredible locations such as this, Bayan Zag, also known as the Flaming Cliff, because they face to the west, and when the sun sets, they just light up bright reds and oranges, even brighter than you see them here. And at this location, Andrews and his team, again in the 1920s, discovered some of the most iconic dinosaurs ever. Velociraptor was first found here, as was Protoceratops and Oviraptor, some more well-known dinosaurs. And also the first evidence of dinosaur eggs was found at the Flaming. Their team also found some less spectacular fossils like this, Panakasaurus. Probably the most apt name for any dinosaur that I've ever heard, because Panakasaurus means plank lizard. And this is really the roadkill of the fossil world. It's just completely pancaked, flat. That's an ugly fossil. If I can orient you to it here, it's kind of looking at me. There's the eyeball, there's the snout. You're looking at the skull from the left. Fortunately, this is not the only specimen of Panakasaurus that was ever found. It was the first, but it's not the only one. More and more specimens were discovered, actually quite a lot, to where it became the best known armored dinosaur from the Asian continent. And scientists soon recognized that it belonged to a group of dinosaurs called the Ankylosaurs. These are armored dinosaurs. They're the walking tank of the dinosaur world. They have armor all over their back. They have shields of armor and rings circling their neck. They have these bony protrusions from their skulls and their jaws. And many ankylosaurs, including Panakasaurus, also had a club on the end of the tail to use as a weapon against attack. Despite all of this, one other thing. Some of these ankylosaurs had bony eyelids. So the eyelid was even bony. That's how armored these dinosaurs were. Despite all this armor, they had about the puniest complement of teeth that you could ever see. You're used to seeing dinosaurs with big, sharp teeth, and these had tiny little leaf-shaped teeth, which was always interpreted as allying these dinosaurs with plant eaters. Well, there's one locality in Mongolia that the rocks are the same age as the Flaming Cliff that Andrews. And this place, you can see why it's named what it is. These are the Camels Hump at Uka Tolgod. And it was there in 2005 that the team from the American Museum made a discovery of another Panakasaurus skull. This is not really that exciting because it was the umpteenth Panakasaurus skull. We already have a lot of skulls belonging to this species. And so it sat from 2005 until about 2014 in a plaster jacket, like the one you saw us making, in a cabinet at the American Museum. And it was in 2014 that I was given the opportunity to work on this particular specimen, Panakasaurus. So technology had evolved quite a lot by then. And so the preparator who worked on this specimen said, why don't I CAT scan it for you first, see what's in that big block. This big block here, I could just about get my arms around it, probably about 150 pounds of solid rock. So it's a big block. And our preparator CT scanned it, and he called me up and he said, bad news. I'm looking at the CAT scan. There's really nothing in there. There's some little blobs, but pretty much what you have is rock and some more rock. Now, he had run it through a regular medical CT scanner, the same that you might go for if you had a chest pain or something like that. He ran the specimen through there, and this is what we came up with. He said, do you want me to go ahead and try to prepare it mechanically to get the fossil out, to get in there out of the rock? And I said, yeah, sure. What does that entail? He said, well, if there's a fossil in there, we'll find it, as long as you don't mind the discovery hole. Discovery hole, what's that? He said, it's when I find the fossil, I actually bust through it and create a little damage. I said, okay, I don't mind the discovery hole as long as you can find a fossil. Well, sure enough, there was more than just a fossil in there. Probably the best skull of Panakasaurus and one of the best skulls of an ankylosaur ever discovered. The reason why it wasn't showing up in the CAT scan is because this bone, over millions of years, even though it's a different color, it had acquired the exact same mineral density as the rock that was encasing it. And so medical grade CT scanner couldn't tell the difference between rock and bone. But once you start preparing it mechanically, you can tell the difference. And I need to orient you a little bit to what you're looking at here, because now you're looking up at the bottom. You're looking up at the chin of Panakasaurus here. And there's a three-stage process that I'm showing here, where we started to expose the rock by chipping it away. Here you can see the neck vertebrae. And this arch right here is the lower jaw. So it's just as if you're looking up at the chin of this animal. What puzzled us at first, as our preparator started to work on this, was this area right here. We didn't know what to make of this bone. We thought maybe Panakasaurus had swallowed something, or maybe that was another bone of another organism that had kind of washed into close proximity with this Panakasaurus. But we decided to continue preparing it, and this started to emerge, and we were finally able to remove it and recognize it as the branchial arches of Panakasaurus. We were looking at for the first time the branchial bones preserved exactly where they're supposed to be, in their life position in an extinct dinosaur. We found out that the branchial bones of Panakasaurus were dominated by this strange shape here. Now, where'd you see that before? It's the arrowhead shape that we saw in the branchial arches of birds. We later found out that this is called a paraglossal bone when it's present in birds, but it had never been described in dinosaurs before. And then behind that were remnants of arch 2 and arch 3 and a little bit more of arch 2. But because these bones were preserved in perfect alignment to where they were in the living organism, we were able to deduce they were probably held together by a central cartilage that had wasted away. So just imagine all the bones of your Thanksgiving turkey being right in their place, but all the soft tissues kind of getting out of the picture, and that's what we see here in branchial arches of Saurus. So this was really exciting. We did some more CT scanning of this set of branchial bones. We made this 3D model so we could explore literally every nook and cranny of these bones. So this is the 5-year-old me, many years later, putting names on all these little grooves and all these little bumps. Every one of these little holes has an artery going through it, and every one of these little grooves has a muscle attached to it, purpose in the organism. And this is a great opportunity that we had that wasn't available just a few years ago to make 3D models of the bones and explore the structures that are otherwise covered up by rocky matrix. And this allowed us to make a really good working model of what the branchial arches of Panakasaurus look like, and to put it in context of the actual skull. Now remember, in reptiles, these are bones that support the bottom of the floor of the mouth and the tongue, and they're attached to muscles of the throat. But we didn't really know what to make of them in Panakasaurus because they didn't look like anything else that anybody had ever seen before. Well, this arrowhead shape, we had seen that before in birds. And so we started looking at the birds' branchial bones. Since birds are known to be closely related to dinosaurs, we started looking at birds to try to get at the function of the branchial bones in Panakasaurus. And while that arrowhead shape was there, both of them, maybe a little bit bigger overall in Panakasaurus, this back part, kind of this boomerang part, was not really working. It didn't fit into the model that we were finding in Panakasaurus. And so I looked through just about every set of illustrations of bird branchial bones, every reptile that I could think of. We're looking at lizards, snakes, all the reptiles that we could compare it to. And we're just coming up blank with this pattern. There was nothing that really resembled this. Again, the branchial bones are as unique as the individual species that they belong to. And then one day I was literally sitting at my desk, and I had gotten tired of looking at all those pictures of reptile branchial bones. Just turned my head to the left, and this book was sitting right on my stack of papers there. And on the cover of this book is X-ray of the floor of the mouth of a terrestrial salamander. And sure enough, right there, that exact shape that we saw in Panakasaurus. So this started to look really funny to us because Panakasaurus it's an anatomical mosaic. It's like the front of a bird branchial bone stuck on the back of a salamander branchial bone. And where are these triangular branchial bones of the salamander? Well, they might have a home right back here. So Panakasaurus had a really strange combination of very primitive features, like you see in this salamander. It's almost as if it had gills. And very advanced features, like you see in modern birds. Now, again, what these bones are good for is attachment of muscles. Specifically, the tongue musculature attaches to the kind of front part of all these bones. And what can you do with your tongue? You really can only do a few things with it besides taste. You can stick it out, you can pull it back into your mouth, or you can move it around from side to side. That's about it. But we started looking at other animals that have that use their tongue a lot, and they tend to have really robust branchial bones with a lot of muscle scars on it, indicating that the muscles are attaching there and moving the tongue around a lot. So we think that Panakasaurus had a muscular and a very fleshy tongue, and also remember what kind of teeth it had. Teeny tiny, the puniest teeth in the dinosaur world. So we think that Panakasaurus used its tongue a lot more. Lingual processing, or using the tongue, was really important to Panakasaurus in terms of feeding, much less so than chewing. This is also supported by independent research that shows that Panakasaurus had this kind of spiral movement to its jaw. It actually kind of had a scissoring movement, not a chomping movement. And so lingual processing combined with that scissoring movement was probably important to the feeding mode of Panakasaurus. Now, it's always been branded as a plant eater. All the ankylosaurs and actually all the ornithischian dinosaurs to which this group belongs have always been considered to be plant eaters. But we suggested when we published on this that Panakasaurus may have supplemented that diet with insects because of the strength of the tongue muscle and the fact that we see long tongues and robust branchial bones in things like anteaters that are primarily insectivores. And also, you go back to that locality, Ukatolgod, there's abundant evidence of insects in the form of boring, where the insects have left their traces, and little burrows that contain millipedes. So it's possible that Panakasaurus, always considered plant eater, might have supplemented its diet with insects, or maybe even subsisted primarily. We were also able to put Panakasaurus in its rightful place in the evolutionary tree. So if you remember our tree of common ancestry, we can place all these branchial bones of birds here. We know that they all belong together because they're all derived, well, they're all birds and they're all derived from common ancestor birds. Panakasaurus belongs way down here. It's very, as dinosaurs go, it's very distantly related to birds. So to find this pattern, this paraglossal bone with the arrowhead-shaped configuration, very characteristic in a dinosaur so distantly related to birds, means that the framework to build that bone probably characterizes all of dinosaurs. So all of dinosaurs had the capacity to make a branchial apparatus that looked like this. Whether or not they did it remains to be seen. We don't have the fossils that show it yet, but at least now we know what we're looking for. We have these are some of those pencil-sized bony elements. Are we just missing the rest of it? Or was it cartilaginous? We don't know, but more explorations in museum collections and in the field will surely tell. So what about us? Feel your own neck. I don't feel any gill slit. Feel the bottom floor of your mouth. I don't feel any sharp protruding bones like we saw in Panakasaurus. So what became of the branchial arches of human beings? Well, think back to what Darwin said. Their evidence is in the embryo. So if we look at this little human embryo, about 4 weeks old, you can see here's his head, tail. These little buds will become the lower limb, upper limb, leg, arm if you want. This big bulge here is going to become the heart. And right here behind the eye, just like in the fish in your aquarium, there's the gill arch. This first arch should become the jaws. Remember, we did that a while back in evolution. And then the next few arches, what becomes of them? Well, I've said if you feel under your tongue, you don't really feel a big arrowhead-shaped bone like Panakasaurus. But you should feel some bone, and you should feel some cartilage. So your Adam's apple, your voice box, or for the anatomy learned here, the laryngeal prominence of the larynx, right? Those cartilages you can feel right under your skin. And then if you work your way up from your Adam's apple, you might feel buried under there, under your tongue, a little bone. That's called the hyoid bone. These are the gill arches, or the bones of the gill arches in your own body. And this color-coded view shows you that first arch becomes your upper and lower jaws, and the ones behind them, in a fish, just remain gill arches. But what we see in us, first arch becomes the jaws, and for that matter, some of the little bones in the ear. And then behind that, we don't have gills. We've got a hyoid bone and the cartilages of the larynx. So this is what happened to the branchial arches in us. Now, why is this important? Why is this important to us? Why is it important to students of medicine? Well, things can go wrong with the development of these arches. One classic example is a failure of the first arch to form a reasonably developed jaw and the ear bones that are related. This is an example of a first arch syndrome. This one's called Treacher-Collins syndrome. And if you saw the movie Wonder, or if you read the book Wonder, you saw the main character in that, Augie, had this appearance. He had downward slanting eyes, small jaw and chin, and he had difficulty hearing, you might remember, and his ears were misshapen and not what you expect them to look like. So this is an inherited condition in which some of the bones and tissues of the face don't develop. It's the first arch that's not developed. These patients are of normal intelligence because nothing's wrong with the development of the brain, just the first arch that's leading to this characteristic. And you might remember, if you saw that movie, that Augie said that he had several surgeries to correct the growth of these parts, again, derived from the first arch. Another situation in which the development of the arches becomes clinically important to doctors and to medical students is the occurrence of what's called a branchial cleft fistula. So some people have a little hole in the side of the neck that will ooze out just a little bit of fluid, and it might not bother them for a while. You might not even recognize what it is. But if they come under clinical observation for it, you can inject a little bit of metallic contrast, or like a little bit of dye, paint, into that hole, and it will follow a track all the way up into the mouth. This is what's known as a branchial cleft fistula, and what it is essentially is the retention of a gill slit from the embryo. Think about the relationships that we talked about in the fish breathing. The fish takes in water through its mouth, and it passes down through where the neck would be, and then out a slit in the neck. A retention of the same structure in a human being leads to a branchial cleft fistula. Now, this is something that can become infected, but it's relatively easy, certainly compared to the previous situation, to correct surgically. So I thought I would close with this quote from Darwin, again from The Origin of Species, to bring it all together, because he said, "We may cease marveling at the embryo of an air-breathing mammal or bird having branchial slits and arteries running in loops, like those of a fish which has to breathe the air dissolved in water by the aid of well-developed branchiae." In other words, Darwin was saying that the evolutionary history, or the ancestry, of every organism is essentially written in the embryo. And so, just to conclude, I hope I've been able to show that human anatomy and dinosaur anatomy aren't all that different. even though Panakasaurus hatched from an egg 75 million years ago, and we were all born in the last, well, 209 years, we all share common ancestors, and that common ancestry is really what is demonstrated by our shared similarities. Our shared similarities demonstrate our common ancestry. And last, some human diseases and disorders make more sense in the light of evolution. And so I think it's important for medical students and doctors alike to learn about principles of evolution, because it can lead to better patient care. If you're interested in this topic, you can't really do a lot better than Your Inner Fish by Neil Shubin. Neil Shubin spins an awesome tale that touches on some of the same things I've talked about today in a lot more detail. I also recommend Dinosaurs of the Flaming Cliffs by Michael Novacek. This book describes the discovery of Ukatolgod and even mentions Panakasaurus in there, a little appearance. And The Origin of Species is eminently readable. I really recommend reading that. I listened to the audio book for it. It was expertly narrated. Sounded a lot like you, Mr. Darwin. Or maybe you have a couple of books in your basement and you can start, as I did, with those. So with that, I thank you for your kind attention, and I'm happy to take any questions. Sure.
The anatomy of the human foot in any other species. It seems, to my limited knowledge, the only contrast that humans have to every other species.
So your question is about the anatomical contrast of the human foot versus every other species of primate.
Right. They have hands, your arms, everything else is similar, but...
Absolutely, yes. And so I would say that's a relatively easy comparison to make because primates are so close to us. You have to really stretch, as I tried to do here, to talk about the bones of something so distantly related as dinosaur. If you're talking about primates, that's a lot more closely related to us. And so the degree of something that might seem a minor difference becomes really important. It is absolutely true that humans have a very unique foot, and that's part of what allows us to have a bipedal gait. And so all of our toes face forward, whereas most other primates have a big toe that's splayed out just like a thumb. In many cases, it's prehensile, it's grasping. In species that run around in the trees, they're grabbing onto the trees with both hands and feet. So you're absolutely right that the architecture of the human foot is totally unique, and it's one of the most important things in the evolution of bipedality as kind of a unique human behavior. For the anatomy students in the room, even more specifically, there is a bone called the medial cuneiform bone, and that's kind of the keystone from which your big toe is attached. That bone is like a perfect square. It's like a perfect cube. And so there's not a lot of movement that would be permitted there. Why you have a hard time grasping things with your big toe, but our primate cousins have a little more flexibility.
So it is unique to humans, the human foot. There's not quite anything that in other species you could look at and say, I see the evolution.
I see what you're saying. Yeah, so if we're looking at just living organisms, our closest relatives are the great apes, chimpanzees, gorillas, and so on. And their feet show a degree of flexibility in that big toe joint that we simply don't have. However, if we look at that evolutionary tree, there are intermediate forms between them, not living on Earth anymore, but extinct and sharing a more recent common ancestor with us than do the living great apes. And those show degrees of progressively getting more square, more forward-facing big toe. But then again, when you're looking at an individual fossil, it's hard to tell how important just a degree of difference is. So that's why I think it's been important for scientists who look at these transitions. Where exactly do you draw the line? How do you know that this extinct hominid was walking on all fours and this one was walking?
One more, if I can.
Yes, please.
When you pick up a fossil, is the fossil still a bone, or is it I can't think of the term where it becomes a fossil?
Yeah, it's a great question. It's a great existential question for the organism that became the fossil, and when does it stop being a bone and when does it start being a fossil? There are different modes of preservation of fossils. The kind that I was talking about here, and probably the most common kind, you have a bone is buried in sedimentary rock, preferably very quickly, and as water trickles through that rock over millions of years, it replaces the existing minerals in the bone with whatever minerals are prevalent in the rock. And so the bone essentially turns to stone. Petrification of wood is kind of along the same lines as this. But you can have an insect that's trapped in the sap of a tree and become fossilized as amber. Now that's a fossil, but it hasn't undergone that mineral transition that I was talking about with the bone. There's different modes of fossilization.
Yeah, and that example that you had gave earlier about this encasement, and you took a CAT scan and you couldn't tell bone from
Yeah.
So, but as he chipped away at it, how is it he didn't chip away and it become, how did he wind up having a bone when it was almost bone and fossil was the same?
Yeah, so that's an example of the bone became completely, the term is permineralized. The minerals of the rock leached into it and made the density exactly the same. It was not completely changed though, because you could see the color difference and you could see the structure. Yeah. Yes, absolutely. It's on a spectrum like that. And there's also a condition called subfossilization. So there are some fossils that are maybe only 2,000 to 3,000 years old, not millions of years old. They haven't had the time to turn to stone, so they still feel like a bone, a dry bone.
Hello.
Thank you. Hey.
So first of all, this is probably going to sound really nerdy, but you're a personal hero of mine. I read The Flaming Cliffs. I loved the book with Dr. Bennington's class, and You're in a Fish. So I'm familiar with both work. Very interesting that you were at Ukatogod.
Well, I didn't write those.
Well, you were on the expeditions, which makes you cool in and of your own right. So congratulations. Just take the win.
Thank you.
So basically what I was wondering is there are morphologies, like you said, with the pharyngeal, the gill arches, how they differ in different organisms. And you said that if it was in this ankylosaur, it would be featured in other dinosaurs because of how it is morphologically similar to ancestral traits. So basically I was wondering, is that the same case with feathers? Because there are ornithischian dinosaurs with feathers. That have been found, quilled animals like Psittacosaurus has quills. Does that mean that all dinosaurs, including the ones in Saurischian dinosaurs, would both be covered in feathers?
Yeah, great question. So the short answer is it's complicated. And anytime you find one of these features outside the expected clade or outside the expected group, it's tempting to just kind of pull all that down to the most recent common ancestor of both of those, because that goes on the assumption that evolution is rare and that one origin of feathers is more parsimonious than multiple origins of feathers.
Right.
This goes right to Darwin's theory that we've got common ancestry for this. It didn't just appear multiple times. That being said, we're finding out more and more that when a feature is useful, living things tend to hit on it more than once in the course of evolution. So just a photoreceptor, like the eye, has arisen many times in different lineages in different
Right.
ways, because all of those organisms recognize it's important to know light from dark.
Right.
Yeah. So the same may be true of feathers, and I think that would require looking really closely at the quills, as you said, in some of these ornithischian dinosaurs, the kind of peach fuzz that's known now from some basal theropods and things like that. But it'll be really interesting to see what that shows. There are some really wonderful fossils known from China, which are now being looked at with ultraviolet and different kind of light spectrum technologies, and finding more and more of those skin structures and feather type structures. Yeah, this is something that was not possible years ago, but now we can use scanning electron microscope. First of all, we can recognize fossils that include feathers preserved in the fossil record, and look at them with scanning electron microscopes and other technologies like synchrotron and things like this, and then at a really microscopic level, characterize organelles. Actually, melanosomes have been found associated with these fossils, where you can extrapolate the color that some feathered dinosaurs may have been.
Okay. Yes. Thank you. It was very interesting. And I'm just wondering, you were focusing on the brachial bones and the gill slit. What other parts of the anatomy have been compared between dinosaurs and humans that have shown a common thread like those other things?
Thank you. Yeah. Well, again, you have to go really far back in time to find the common ground of the common ancestor that gave rise to both us and dinosaurs. We're both at the tips of those branches, so to say, and we've had a long time to develop our own special features. But if you get down to what unites us and dinosaurs, lots of things. For example, we have a backbone made up of multiple vertebrae. We have that pattern: one bone here, two bones there, movable supports in the wrist. And so some of these basic things characterize dinosaurs, humans.
Hey, over here. So I had a question about, essentially, since I know you're mentioning hair-like filaments, and since we're basically from the synapsid lineage versus the diapsid like dinosaurs, how is that transition between the acquisition of some genetic characteristics that can, say, form like the vestibulocochlear apparatus, then being co-opted with those bones to create, the middle ear apparatus, versus the way that birds and non-avian dinosaurs use a completely separate way, but I mean, would you say would still have the same genetic lineage? And so how, in that whole idea of exaptation, how is that transition between developing the fertile ground, to then growing into the different kind of organ systems that you get?
I think I understand your question. You're asking about kind of like the genetic underlying mechanisms that are responsible for what we see in terms of the morphology of these animals.
Right, because they're both, say, hearing using similar things, but the actual anatomic method by which they hear are so vastly different.
Yeah, and so this is why that's more complicated than the other questions that we've had tonight too. This is why total evidence analyses are so important. Analyses that take into account not just morphology, but when it's available, DNA sequences and soft tissue morphology and cellular features and all. And it gets more complicated than that, because I tried to paint a picture here where you say, okay, well, the final product might be different, but at least we know the development. It's widely thought now that development can evolve too. So we we used to think, okay, we can rely on the developmental pathway, that these are the steps that will take to give us whatever organism it is, or human being. Sorry? Right, right. But now if that developmental pathway is subject to natural selection, then all bets are off. That the pathway could change completely. We might have an incomplete picture of that evolution. Certainly, yeah, and there are lots of heritable conditions that are dominant. They're dominantly inherited, and yet they're deleterious to the person inheriting them. So there's also a lot that goes on in at the cellular level in terms of gamete formation, crossing over, and you get linkage disequilibrium of individual gene mutations associated with another chromosome. So I think it's a complex picture that you never really see the whole picture if you're just looking at one side of it. If you're just looking at the dinosaurs, okay, that's one thing, but widen it out and look at birds, not just birds, but at the genetic level of birds. It just kind of depends on how you focus your lens. Thank you.
Yeah. Hi.
Yes, sir.
Yeah, you were talking about transitional forms before. I've read that there's a lack of transitional forms in the fossil record. That's true?
Well, each transitional form we find is one more transitional form. It depends on how comfortable you are or how comfortable the scientist is with that gap. When I showed the idea of common ancestors, essentially, any fossil that's discovered in the fossil record is placed in that family tree at the tip of one of those branches, and the ancestor is purely hypothetical because we have the fossil, so we don't want to build that straight marching, from the gorilla up to the human being. So we put the new fossil at the tip of one of those branches, and the ancestor becomes hypothetical. So the gap will always be there, but it's always getting smaller and smaller with each fossil that's found. There is a temptation to say, well, if you fill a gap with the transitional fossil, now you got two gaps, one on either side, and so you're not really solving the problem. But you are slotting in each individual new species that's discovered somewhere on that phylogenetic river. Does that answer your question?
Yes, and thank you for the great questions tonight. We have to close off with questions, so please join me in giving Rob Hill a nice round of applause here.
Thank you.